Epilepsy is a disorder characterized by recurrent spontaneous seizures. Seizures are a result of too much activity in a part of the brain at one time. This hyperexcitability is merely a symptom of seizures, and the exact causes of the different types of epilepsy are mostly unknown. Most anti-seizure medications work to restore the balance between excitation and inhibition in the brain, although some patients do not respond well to this type of treatment and might therefore have a more severe or complex dysfunction. Glial cells outnumber neurons in the brain by about 4 to 1 and are thought to be just as important, if not more important, than neurons. Astrocytes are a type of glial cell that helps regulate intracellular fluid in the brain by maintaining ion and neurotransmitter homeostasis. As you can see in the picture, astrocytes achieve this by extending their processes near neuronal synapses and take up any excess neurotransmitters that are floating around. Astrocytes can then break down these neurotransmitters and recycle the components back to a neuron, or astrocytes have the ability to release their own transmitters to further alter synaptic activity. If these astrocytes are changed or damaged in any way, something called astrogliosis occurs, which is the abnormal proliferation of astrocytes. This can lead to glial scarring, as you can see in image C, and has also been implicated in the onset of seizures. Previous experiments have induced astrogliosis in model brains by applying some type of trauma, although in that methodology it is unknown whether the resulting seizures were from the trauma or the resulting astrogliosis. The question that is being investigated here is whether astrogliosis precedes and is therefore a cause of neuronal network hyperexcitability, or does astrogliosis result from seizures and then worsen the hyperexcitability? To answer this question, beta-1 integrin was conditionally deleted in only radial glial, which causes chronic astrogliosis throughout the brain without causing other brain abnormalities, the destruction of the blood-brain barrier, or pronounced inflammation. GFAP was used to visualize the glial cells in the cortical gray matter of beta-1 negative or knockout mice and control mice. The images on the left represent the area of glial cell coverage in green, and the bar graph to the immediate right shows that there are significantly more glial cells in the knockout mice compared to the control. The data from the Western blot for GFAP in the top right corner confirms there are higher levels of GFAP in the acute slices of knockout mice than the control mice, where the bottom graph below is the quantification of the Western blot data. The figures at the bottom of the slide reveal that microglial activity is also increased in the beta-1 knockout mice, suggesting that microglia are activated as a result of the astrocyte activation. Together, this data supports that the conditional deletion of the beta-1 integrin causes progressive reactive astrogliosis. Figures A, B, and C are looking at EEG recordings in control and knockout mice. In Figure A, the knockouts have multiple interictal seizure spikes, while the control does not. In Figure H, interictal spikes in knockout mice increase in frequency with age. No seizures were recorded in the control mice over the course of this experiment. Figure B shows a seizure event in a knockout mouse compared to normal activity. Overall, mice with chronic astrogliosis developed spontaneous seizures, while controls did not. During the experiment, implanting electrodes for EEG analysis caused some degree of tissue damage in the cortical gray matter. As shown in figures A and B, the mice were placed into categories of no damage, mild damage, or severe damage based on histochemical analysis. The mice in the different categories of damage had no significant differences between them in regards to seizure activity, as shown in figure C. This suggests that the focal gliosis from a localized acute injury did not cause seizures, indicating that chronic astrogliosis was the main cause of the development of spontaneous seizures. Here, figures A and B show differences in resting membrane potential and input resistances. The neurons in the knockout mice were depolarized and had higher input resistances, although input resistance was partly dependent on age. Both are important because abnormal changes in neuronal excitability are often associated with epileptic seizures. To see if there would be differences in seizure activity in controls versus knockouts, magnesium was decreased in the system, which is known to cause seizures. As is shown in figures D and E, the time period between spikes and seizure activity in controls was much longer than in knockouts. This was repeated using a different type of beta-1 integrin knockout that targeted only neurons rather than glia. No significant differences between the neuron nex-Cre knockouts and the controls were found, further showing that changes in astrocytes rather than neurons are what causes the increased hyperexcitability. Going off of the previous slide, increasing current caused more action potentials in knockouts, as seen in figures G and H, indicating knockouts had lower thresholds. Figure I is raw data showing that as stimulation increased, knockout mice generated larger excitatory responses. This is showing that the knockout mice are experiencing neuronal hyperexcitability. Next, the Western blot analysis in figure J was used to see if the AMPA receptor expression had changed in the knockout mice. AMPA receptors are important because they mediate fast synaptic excitation and are correlated with higher levels of GLU-R2/3. An increase in GLU-R2/3 protein expression is known to be a contributor in seizure generation, but because this protein decreases in the knockout mice, this idea is excluded. Figures A, B, and C show comparable levels of GLT1 and GLAST between knockouts and controls ages 4 to 6 weeks. Figure A shows no difference in GLT1 distribution for controls and knockouts. The Western blot in figure B shows GLT1 levels were lower in the knockouts, while figure H shows GLAST levels were comparable between knockouts and controls. Blocking glutamate transporters with inhibitors TBOA and DHK gave the data in figure D and shows that glutamate taken up was greatly reduced in the knockout astrocytes. The Western blot in figure E shows glutamine synthetase was determined to be in comparable levels for knockouts and controls. These data support that reactive astrocytes in knockout mice impairs glutamate homeostasis, where increased extracellular glutamate concentrations can contribute to neuronal excitability. Figures B, C, and D are quantifications of data collected from the Western blot in figure A. The data from figures A and B form the ratio between KCC2 and NKCC1 as shown in figure C. As the mice age, there is a steady ratio among the control, but decreases are found as the knockouts age. This data suggests that the neurons in knockout mice may have adopted an immature phenotype. Figures A and B recorded EEGs from 6 to 8-week-old mice while blocking NKCC1 in vivo with bumetanide, where in figure A there were no seizures in the control and seizures were only eliminated from a subset of the knockout mice, as others remained unresponsive to the bumetanide treatment. Overall, this paper was extremely thorough and the findings were backed up numerous times with a variety of experiments. There were an adequate number of controls to compare the experimental data to, so the researchers could say with near certainty that the astrogliosis was directly correlated to the increased epileptogenesis. One minor critique of the paper is that the data is sometimes difficult to follow due to the mass clumping together of graphs and images that may have been better off placed into separate figures. Some important data, like the comparison of the beta-1 knockout in glial cells versus the beta-1 knockout in neurons, seemed hidden in another figure. Although this is a valid critique, the disorganization might be a result of space and figure limitations placed upon the researchers by the journal they submitted to. Epilepsy is the most common chronic neurological condition, and yet so much is still unknown about the differences in etiology and presentation of the disease. 33% of effective patients do not respond to drug treatments, which means there is plenty more research that still needs to be conducted in hopes of developing new and more effective treatments. Assuming that current treatments are effective for a patient, there are numerous side effects that are associated with them. This image represents some of the side effects that can occur with anti-epileptic drugs, including psychotic symptoms, tolerance, and worsening of symptoms. This research, conducted by Robel et al., could be the first step to identifying a different cause of epilepsy, which could lead to the creation of a more specific treatment with fewer side effects. Gene-targeted therapies might also be feasible if a gene sequence relating to astrogliosis is able to be targeted in human patients. It is also important for future research to examine the possible role of epigenetics in astrogliosis and, therefore, epileptogenesis.